Transcription is the production of RNA to DNA. DNA is the template strand. The produced RNA is mRNA, which contains the information to make a polypeptide. The template strand contains a core promoter region, which is a non-coding region located upstream. It also contains a coding region located downstream that will code for one gene at a time. Initiation is the first step of transcription. RNA polymerase is bound to the start codon so that it knows when to begin coding. The -35 and -10 boxes are in the core promoter region. The -10 box contains adenine and thymine residues called the TA-TA box. The promoter signals the beginning of transcription by being a recognition site for transcription factors to bind to activating RNA polymerase. Elongation is the next step. RNA polymerase moves along the template strand in the 3' to 5' direction, making the complementary strand longer. At the end of the molecule, transcription has to be terminated with a terminator sequence. It defines the end of a gene, allowing the RNA transcript to dissociate from the DNA. Eukaryotic mRNA is in the nucleus, but must travel to the cytoplasm to arrive at ribosomes where the proteins are made. In order to do this, it must undergo post-transcriptional modifications. Splicing removes unneeded old sections of mRNA called introns. To do this, it utilizes an enzyme known as a spliceosome. Exons are the good parts of RNA and are pasted together. A 5' cap is added at the beginning of the first nucleotide to protect the transcript from being broken down. It also helps the ribosome attach to mRNA and start reading to make a protein. Another mechanism that protects the mRNA is the poly-A tail. The poly-A tail is added to the 3' end of the RNA and allows proteins to fire off while nucleic acids are destroyed by digestive enzymes. It serves as a distraction mechanism and makes the transcript more stable as it is exported through the cytoplasm. The transcription factors needed to initiate transcription can either be general transcription factors or regulatory transcription factors. Regulatory transcription factors regulate the rate of the transcription of nearby genes. They can be used in activation or repression mechanisms. Small effector molecules may modulate regulatory transcription factor activity as well as response element, which are upstream near the core promoter region. Regulatory transcription factors have structural motifs that are the helix-turn helix, helix-loop helix, zinc finger, and leucine zipper. If it is regulating as an activator, it is increasing the rate of transcription. An activator protein binds to the enhancer, which is upstream near the core promoter, and RNA transcription is increased. If it is regulating as a repressor, it decreases the rate of transcription. A repressor protein binds to a silencer region upstream of the core promoter and RNA transcription is inhibited.